Okay, everyone, welcome back to Chapter 3 lecture of our Biology 1010 series. This lecture is going to cover cells, what cells are, what are inside cells, what are the things that make up cells, how do cells work, and the things that make cells different from one another. So let's go ahead and get started with that. So what is a cell? Well, a cell is often referred to as the smallest unit of life. A better way to put that is this more fundamental unit of life. They're not necessarily small. So cells can be quite small, microscopic, like bacteria and things like that, or they can also be quite large, things like eggs. An ostrich egg is a single cell. So they're not necessarily small, more of the most fundamental um, unit of life, the most fundamental unit of living things is considered a cell. Now, what is a cell and what makes a cell a cell? Well, a cell is the most fundamental thing that can do all of those functions of life, all 5 of those functions of life that we talked about earlier in this lecture series, things like reproduction, growth, and development, um, things like making energy use, and things like that. So a cell is the fundamental unit of life that can do all 5 of those things. Now, every living organism on the planet is made up of at least one cell. So you can be a single-celled organism like this little bacteria here, this amoeba, or a multicellular organism like us or a frog or something like that. Now, biochemical processes inside cells happen all the time. They're having biochemical reactions that make energy, that reproduce DNA and things like that. And all of those things that are happening inside of this cell is what allows life to be life. Cells are capable of doing all of those different types of reactions that life requires, and that's what makes a cell a living organism. They're the smallest, simplest, most fundamental unit of living things that are capable of doing all of the traits of life and carrying out those basic functions, chemical reactions, and things like that that all life needs. Most cells are too small for you to be able to see without a microscope. So if you refer back to our previous lecture series on atoms, atoms are extremely small. You can't see them. Proteins, viruses, and most bacteria are going to be too small for the human eye to see. Most plant cells and animal cells are going to be too small for you to see. And right about the edge of frog eggs, some of the large animal cells and things like that, you will start to be able to perceive with the human eye. So the vast majority of living organisms are single-celled microscopic organisms, and then we just can't see them. So anything larger than that, frog eggs and ants and larger organisms are made up of more than one cell. We can see with our naked eyes. Cells are most of the time going to be way too small for us to see with our own eyes. So to see them, we have to use a microscope. Most commonly a light microscope you can see up here. This is the most common type of microscopes that you find in labs and hospitals and schools and things like that. It visualizes the vast majority of things that humans would need to see underneath a microscope. Most bacteria, most cells and things like that you can see underneath a light microscope. Electron microscopes allow you to see the insides of cells. They can allow you to see down to the atomic level as well. So we can see very small things with those electron microscopes. They're very expensive. They're very large. They take up a lot of space. They're difficult to use. So you need to be a little more skilled on how to use one of those machines. So they're a little more specialized, so not a lot of places are going to have those. So you need higher magnification, which the electron microscope can provide to see these much smaller organisms and these much smaller particles and things like that. So our microscopes that we use on campus are going to be compound light microscopes. So that means that they can see the vast majority of most bacteria all the way up into right around the end of frog eggs. And anything bigger than that, you're not going to be able to really see. It's going to be too big to fit underneath here. You don't really need it. You can see it and make a lot of detail out of it. with your naked eye. You don't really need a microscope at that point if it's bigger than that. You can see it, but you can get a lot of detail just by looking at an ant up close. And with your eye, you can see it. You don't really have to have a microscope. Well, anyway, the microscopes that we have on campus look like this. You can see the parts of them. And they're put together to allow you to make microscopic changes and magnification changes that compound on top of itself. So these ocular lenses here have a magnification power of about 10. And the light comes through them down through the microscope, up to the microscope and up to the lenses here. And the objective lenses have magnification power as well. And they change. So 10, 4, 10, sometimes 100, 1,000, things like that. It just depends on the microscope in question. Well, anyway, this magnification power here compounds on the magnification power here. And this is why they get the name compounding light microscopes. They add up on top of one another to give you a much higher magnification power than one of them by itself, so it compounds. A confocal light microscope is another type of microscope that allows you to see different things inside of the cells, which are very cool. It uses different wavelengths of light, which is neat. And you can focus on little sections inside of a cell or little sections inside of a bacteria or an organism or something like that with the different types of wavelengths of light. You can also use different types of dye that stick to different types of structures inside of the cells. And then what you can do is you can change the light that's coming through to allow one of those structures to fluoresce like a UV light. If you've ever been to a laser tag course, you know that white turns like very, very, very fluorescent purple underneath a UV light. And it's the same kind of concept that's going on here. They don't look like anything fancy until they're exposed to UV light. And this type of microscopy allows that to happen, which is pretty cool. So, transmitting electron microscopes, these are really cool. These guys use filaments of gold, little tiny small gold particles, to see what's going on inside of these cells. So what they do is electrons are significantly smaller than light beams. So light beams are what makes light microscopes work. They work on light beams. Light beams are very large. Electrons, on the other hand, are very small. So electrons can make it's the pixel concept if you're familiar with how that works in TVs or cameras or something like that. Bigger pixels, the less quality image you get. Smaller pixels, the higher quality image you get. So whatever you use to take the picture is, in the case of light, is a big pixel, whereas our electrons are smaller pixels. With the electron microscopes, you get a much better picture. Well, anyway, the electron microscope uses electrons to take its picture instead of light, which allows much higher resolution. It also allows you to be able to see through the cells with the electrons instead of blocking things like shadows and stuff with the light microscope. So you get much greater detail. You can see much more resolution. You can see through the cells and things like that. It's really cool how these things work. So you get really great detail. You can zoom in to the atomic level in very, very, very great detail. You can see with the electron microscope, whereas you won't be able to achieve that with our light microscopes. Scanning electron microscopes, on the other hand, they just scan the surface. The transmission ones go all the way through. Our scanning electron microscopes are used to deflect off the surface of cells. Now, I think I misspoke a second ago when I mentioned that they use gold electrons. Now, this is the one that uses the gold. This one uses gold foil. It coats the surface of these cells in a little small gold dust, and the electrons bounce off of that, and that's what generates the picture, the electrons bouncing off the surface of that gold. Very, very, very interesting process. Creates a lot of detail at the surface of what a bacteria would look like, of what a virus looks like on the surface. So this allows you to see the protein structures and things like that on the surface of cells. Very, very, very cool. Now, cells can vary greatly in size, and I think I mentioned that already. Bacteria are very, very, very small, and they're about 10 times smaller than the average plant cell or average animal cell. Now, frog eggs are about 10 times larger than the average plant or animal cell. And as I mentioned earlier, an ostrich egg is the largest single cell on the planet, and it's significantly larger than all of these combined. So that's very, very, very interesting on the size differences of cells and how big cells can come and how small cells can be, and they all can carry out every single one of those biological functions that are necessary for life all by themselves, which is really cool. Little tiny things are capable of doing every single trait of life, and even those large giant cells, ostrich eggs, frog eggs, and things are capable of doing that by themselves as well. Now, regardless of the size, every single cell on the planet is going to share some characteristics in common, and this is what makes cells cells. As a scientist, we have to be able to define what something is to be able to classify it. So we have to have a couple of traits, like we talked about with the traits of life, to classify what a cell is and how do we define what a cell is and what's not a cell. So these traits here are what's going to classify a cell. You have to have all of these traits in common to be considered a cell, and all of these things are necessary to carry out all of the basic chemical reactions of life. So without these things that we're about to talk about, you won't be able to be a living organism. You won't be able to do all of the chemical reactions to make your DNA, to make proteins, to make energy that are required to live. So every single cell on the planet, regardless of if you're a teeny tiny little bacteria or a giant human cell or an even larger ostrich egg, you're all going to have genetic material inside of you, both DNA and RNA, and that's a key characteristic here of living cells. They have both DNA and RNA inside of them. Ribosomes, you're going to have ribosomes if you're a tiny little bacteria, or a human cell, you're also going to have ribosomes, and those are going to be where proteins are produced. You have to have proteins to be able to live. Proteins function as enzymes. Proteins allow your cells to be able to do work and to build structures inside of them and things like that. All cells are going to have cytoplasm. And cytoplasm is what, if you like the concept of a water balloon, think about that. Our cell membrane here, which is another characteristic of a cell, is going to be the water balloon itself, the little rubber part. And the cytoplasm is going to be the rubber inside, or the water inside of our water balloon, or the cell membrane. So the cytoplasm is what gives the cell membrane structure. It's what gives the cell membrane the ability to inflate the water inside of the water balloon. If there was no water inside of the water balloon would just be a flat little ball, a flat little blob, and nothing would be able to be done inside of it. No water. And without the water balloon to hold the cytoplasm would just ooze out into the environment, into the world. So both of those things work hand in hand. The cell membrane keeps the water inside, and the water inside keeps the cell membrane inflated and keeps it open. So all four of these things here have to be found inside of a cell for it to be considered a living organism or a cell itself. So that's how that works. Cells are very small for the most part, and the reason why cells are so tiny has to do with how they eat in the surface area of a cell. So cells, when cells get bigger, the bigger you are, the more energy it takes to power you, and this is how this works. If you get, the bigger you are, the more energy it takes, the more food it takes, the more gasoline it takes to power the car to run a larger distance, and that's just how this one works. So cells are very tiny, so they're small. They don't have a lot of it doesn't take a lot of energy to power something that's quite small. Now, if you get bigger, the bigger you get, the more energy that's going to take, up until a certain point. You're going to get so big that you're not going to be able to get enough food or to be able to get rid of enough waste inside of your body at the same time before you can take enough food in, or you're not going to die from having waste build up inside of you. So at one point in time, it's a trade-off. You get so big that your cells are going to be producing so much waste, requiring so much food, that there's no way you can get that much food from your environment, or exchange that much waste, or even exchange that much oxygen with the environment without dying. And that's how that kind of works. It's a very odd trade-off. So most cells are very, very, very small, because as you are, the smaller you are, the more or the less energy it takes for you to survive. So cells can only get so big until that concept of it takes more energy than you can acquire from the environment starts to play in with that. So to keep us going, we have to have oxygen exchange with our lungs very fast, very quickly. So we need smaller cells to be able to do that. If we had really, really, really large cells, those big cells would take so much energy and so much oxygen to power them that they would not be able to exchange oxygen quickly enough with the environment, and then we would die. So it's a very odd trade-off on how this works. So let's go ahead and talk about how cells are divided into different categories in the natural world, and what the three different types of cells are, or two different types of cells, I should say, and the three different domains that cells are broken into. Now, every single cell, regardless of if you're in the domain bacteria, the domain archaea, or the domain eukarya, are all going to have those four things in common that we spoke about earlier. Our DNA and RNA, our cytoplasm, our cell membrane, and then all those other rest of those traits. So they're all going to have all of those things in common, and our ribosomes, they're going to have all of those things in common. So all three of these domains have all four of those traits, and that's what makes a cell a cell. But there are differences between them as well. And those differences are what's going to break them up into these three different types of domains, and what's going to allow scientists to classify them. So, one of the categories down here, domain eukarya, we are eukaryotic organisms. Our cell types are eukaryotic. They have a nucleus, and this is a very important characteristic here, the presence or absence of a nucleus. And this is what's used to really break apart the different domains of cells, prokaryotic versus eukaryotic. And you see these terms here. Eukaryotic, karyotic means nucleus or DNA. The word eu means true in Greek, so true nucleus. Pro means pre, so pre-nucleus. They don't have one. So prokaryotic cells lack a nucleus, whereas eukaryotic cells have a nucleus. You can see that here, and that's a big deal. The other one of those that goes along with it is the presence or absence of membrane-bound organelles. Now, the nucleus is a membrane-bound organelle. Prokaryotes do not have membrane-bound organelles. They essentially are just a membrane-bound organelle. Whereas eukaryotes have a membrane-bound organelles, quite a few of them, and we'll talk about some of those later on. The different types of cell membranes you can see here. Fatty acids, we'll talk about phospholipids and things like that. Non-fatty acid lipids, we'll talk about those maybe in a little bit. And our fatty acid phospholipids again. So our bacteria and our eukarya have very similar membranes, whereas our domain archaea does not. And we might get into some of the differences in between that in a little bit. You can see here our prokaryotes, both domain archaea and bacteria, are very small, very, very, very tiny. Whereas domain eukarya is quite large. So that's one of the big differences, other than the fact of the presence or absence of membrane-bound organelles and nucleus, is how big they are. Prokaryotes are significantly smaller for the most part than eukaryotes, and that's one of the easy ways to tell them apart if you can't use anything other than just a microscope. You can see one's bigger than the other. So let's go ahead and talk about prokaryotes. So prokaryotes are the most ancient form, the most simple form, the smallest form of a cell of living organisms on the planet. We have fossils of bacteria that date to about 3.5, 3.8 billion years ago on the planet. They're very, very similar to what we have now as a species. These guys lack a nucleus on the inside of them, and instead of having a nucleus, they essentially just have a big blob of DNA. Now, a nucleus, as we'll talk about in a little while, is a blob of DNA surrounded by a membrane. These guys don't have a membrane that surrounds their blob of DNA. They lack that nucleus. They don't have a ball of membrane that surrounds their DNA. They have a cell membrane that surrounds their entire cell, and that functions the same way. They're really, really, really small. So they don't really have a lot of space to compartmentalize things inside of them. So there are two different types of prokaryotes. We have bacteria and archaea. Bacteria, the domain bacteria, and the domain archaea. Both of them are prokaryotic. They both have that lack of a nucleus. They don't have membrane-bound organelles, and they're all very, very, very small. Now, eukaryotes, on the other hand, are a little more complex. They're usually significantly larger than our prokaryotes. And they evolved billions of years later after prokaryotes, around 2.3 to 2.5 billion years ago. Our first eukaryotes started to appear on the planet. So a long time afterwards. But eukaryotes are usually, like I mentioned, much larger, and they tend to have a little more compartmentalized functionality on the inside of them. They have things called organelles. And organelles work kind of like your organs inside of your body. Prokaryotes are essentially just a blob that does everything that a living organism needs to. It doesn't really have a liver, it doesn't really have a heart, it doesn't really have kidneys, everything, the one cell just kind of does everything. Now, eukaryotes, on the other hand, they have more like kidneys, more like livers, more like lungs, just specialized little things that are built for transporting food around, eating food, breaking food down, getting rid of waste. They have specialized little structures inside of them called organelles. Now, protist, we talked about the domain protista way earlier, or kingdom protista, excuse me. We've talked about fungi, plants, and animal cells are all considered eukaryotic type cells. Their cells all have those compartmentalized membrane-bound organelles as well as a membrane-bound nucleus. So let's talk about a bacterial cell from the outside in and what's going on the inside of them. So bacteria are prokaryotes, and I mentioned that earlier. They lack a membrane-bound nucleus. They don't have membrane-bound organelles, and things like that. So a nucleus is essentially, like I mentioned, a little ball of phospholipids, lipids, fatty acids that surround the DNA of an organism. It protects the cell's DNA from damage to the outside, or from damage to the DNA from the cell's environment, or from the internal parts of the cells. There's some enzymes and things that like to break down DNA inside of a lot of cells. So it protects them from being broken down. So these bacteria here, they're quite small. They don't have a lot of those enzymes and things like that. So they don't really need a nucleus. They don't really have to protect their DNA. They're so small, they don't really have to do that. So their DNA just kind of floats around inside of them. DNA sticks to itself based on electric charge. So it just sticks to itself, sometimes in a little blob called a nucleoid, or a bacterial chromosome is another term you'll sometimes hear. Well, everything inside of a bacteria floats around inside of the cytoplasm. Now, if you've ever seen fruit jello, you know what I'm talking about. The little jello that wiggles on the table, and it's got little fruit particles inside it floating around inside of it. Now, the cytoplasm is the jello. The DNA, the ribosomes, and all the rest of that stuff is the fruit. Without the cytoplasm for the fruit to float around in, all the fruit would just sink to the bottom of the jello mold and not be able to do anything. So the jello, the cytoplasm, gives the cells space. Like I mentioned, the water inside of the water balloon. It allows stuff to float around. It allows proteins to move from one place to the other because it has something to float through. You can't move a boat across the air because boats don't float in the air, but if you add water, you can move the boat, and this is essentially what's going on. It's the water that allows stuff to float from place to place inside of the cell. So the DNA, the ribosomes, and all of that stuff inside of a prokaryote are just floating around inside of the cytoplasm. They don't have membrane-bound organelles. They essentially are just a membrane-bound organelle. Now, animal cells, on the other hand, plant cells are the same. And it's a little different, but they function very similar. They're eukaryotes. We have tons of membrane-bound organelles inside of our cells, and you can see them all here. They all have specialized functions. Our nucleus protects the DNA. It's essentially just the control center of the cell. It keeps the DNA safe. The endoplasmic reticulum here produces lipids, moves them around, produces proteins, or is where proteins are produced, excuse me. It alters proteins, things like that. Our cell membrane keeps the cell safe. That's prokaryotics have that too. Our Golgi apparatus here moves things around inside of the cells. Our cytoskeleton, our centrioles, and things like that. All these different, our mitochondria makes energy. All of these different specialized structures are called organelles, inside of our cell. Now, plant cells, as I mentioned, are also eukaryote, and they have most of the same membrane-bound organelles in a plant cell that they do in animal cells, but they have a couple of different ones. One of the big ones that you'll probably notice is this thing right here called a central vacuole. Now, this is where the plant is going to store water. Water is way more important to plants than it is to humans. This is how they stay upright. They don't have bones, so they essentially just suck up a lot of water, which makes their cells stay really rigid. This is where they're going to store all that water. Plant cells also have cell walls, and that is something that animal cells lack. It's a very, very, very thick, rigid structure made of can't think of the word right now, made of cellulose. That provides support and gives the plant cell structure and rigidity. So that's another big thing that plants have that we don't have. And the remaining one here is chloroplast, the organelles that allow them to be photosynthetic. This is where photosynthesis is going to occur. We are obviously not photosynthetic. We are not green. We cannot stand out in the front yard and make our own energy. So this is another difference between plant cells and animal cells, the presence or absence of chloroplast. So cells that are eukaryotic are really cool in the fact that I mentioned that they are compartmentalized. And this is what we're going to talk about here, how organelles that are cells that are eukaryotic are compartmentalized. So the very first thing we'll talk about is the cell membrane. Now, every single cell on the planet is going to have a cell membrane. It's going to surround the cell, and it's going to form a barrier between the outside world and the inside world. This is the cell's skin. It keeps stuff on the inside where it's supposed to be, and it keeps stuff on the outside where it's supposed to be. So that's really what's going on here. It keeps stuff in and keeps stuff out. And it keeps things where it's supposed to be. It works the same as the cell's skin. Very, very, very important. If the cell membrane is ruptured, the cell can have some serious problems, the same as if your skin is ruptured. What it also does is it helps regulate things that move in and out of the cell. So things, ions, nutrients, waste products are going to have to move out of the cells through those cell membranes, or in and out of the cells through those cell membranes. So the cell membrane is going to regulate the movement of those things in and out of the cell to help maintain homeostasis. If I need more water, if I need more nutrients, I'll let more nutrients in. If I need to get more waste products out, I'll let more waste products out, and that kind of stuff. So what makes up the cell membrane? Well, the cell membrane is made up of things you've probably heard of before called phospholipids. Phospho meaning phosphorus. You can see that right here. It's the little circle thing, phosphate group. And lipids meaning fats. And this is the lipid part down here. The fatty acid tails. So you have the phosphate part, the lipid tails, so phospholipid. And this is how this works. You can see here a phospholipid is made up of a phosphate group, the little circle part, a glycerol, which is this part up here, the more dark green, and two fatty acid tails. You can see them both down here. This is what makes up a phospholipid. Cell membranes are made up of phospholipids, groups and chains and long chains of phospholipids. But before we get into that, this is how the chemistry of a phospholipid is set up, and this is how cell membranes form. So, cell membranes, or phospholipids, are broken up into two parts. You have the head membrane, the circle part, the head portion, and the tail portion. And you can see that down here, the dark brown, or the little more tan part, the green and tan. So our head part is something termed hydrophilic. It does not like water. It likes water, hydrophilic. It loves water. It's attracted to water. It enjoys water. Hydrophobic, on the other hand, does not like water. Hydrophobic tails, if they come in contact with water, they will be broken down. It tears them apart. They do not want to come in contact with water. The heads, on the other hand, do not mind. They like coming in contact with water. So if you've ever dropped oil in water, you know it forms a ball. And what's happening is all of these heads in the oil, they're lining up to form a protective ball to shield the tails on the inside. So they form a three-dimensional globe structure, like a soccer ball. And the soccer ball, the ball part is the heads that all face out, and on the inside, where the air would be, are all the little tails are hanging down, nice and dry and protected from the water on the outside. And that's what forms a cell membrane. It's the same concept. That little ball theory is how that works. Now, since there's two portions here, we have the top part that's hydrophilic and the bottom part that's hydrophobic. Two different regions of polarity on the same molecule. That's referred to as amphiphatic. Two different regions of polarity on the same molecule. So this is that concept that I mentioned with the ball. You drop the water on the outside, it forms a ball to protect the tails on the inside. Now the problem here is cells have water on the inside, it's the cytoplasm, so we have to protect the cell tails of the phospholipids as well. So the easy way to do that is to just add another layer of lipids on the inside. And you can see that here. You have lipids with their heads facing out to protect from the environment, lipids with their heads facing in to protect the water on the inside, and all the tails are lined up on the middle to stay nice and dry. And this is just how oil and water work, and this is how cells formed. Once these little balls of cells, phospholipids form, things naturally form in the water. This is just naturally how this works. Cells can start to form inside of it. DNA can start to form inside of here, or be protected inside of here. I should say it formed somewhere else. It was protected inside of one of these. This is how cells start to form. This is called a phospholipid bilayer. This is a very interesting little concept. Just oil and water, heads on the outside, tails on the inside. And this is how cell membranes form. So, how this works is cells have very little control over the water that moves in and out of their cells, but they can control ions and polar molecules and some of the larger things that need to move in and out of their cells, but they cannot control the movement of water and lipids and very small things throughout of their cell membranes. So this cell membrane here is said to be selectively permeable. Some things can move through, some things cannot. So if it was all things can move through, it would not be selectively. So the ability to regulate what moves through and what does not move through allows cells to control what goes on inside of them. You need a lot more of these things than these things. Some of these things might be bad. We don't want to instantaneously bring them in, no questions asked. So if we need them, we're going to have to bring them in through work and make sure we bring in the correct ones. Whereas these kind of things just float on through because we need them a lot. So the ability to not have to scan the environment and just bring stuff in when you encounter it is useful, as well as the ability to scan things and not bring stuff in when you don't need them is also very useful. So selectively permeable bilayers are very important for cells. So on the outside of cells, they are also going to have, on the outside of that cell membrane and running through the cell membrane, there are also going to be quite a few proteins embedded in that cell membrane or sticking up on the outside of the cell as well. Now, those proteins are going to do quite a bit of important functions. So some of them are going to be things like transport proteins, and you can see this one here has a hole in it. And this is going to bring stuff into the cell through the environment and take stuff out of the cell from the inside. So the inside here, outside here, stuff in and stuff out. It's going to transport things out of the cell that are too big to just naturally float across that phospholipid bilayer. So stuff's going to float in and out of our cell this way. They sometimes work as enzymes, those proteins, that can allow the cell to do chemical reactions and things on the cell membrane, very important. They sometimes work as recognition proteins, and this is very important. You want to make sure that the cell in a multicellular organism is where it's supposed to be. All the cells inside of me want to be Jonathan's cells. That's important for that. And our body's going to read the immune system, is going to read each one of the little individual cells and make sure that they're Jonathan's cells. The proteins that are on the surface of my cells are what's going to let the immune system know that. Adhesion and receptor proteins are going to be found on the outside of the cell membranes, and that's going to allow cells to stick to different things, allow different cells to stick to them, and that's a very important thing when it comes to transmitting cellular information, sometimes cellular reproduction, preventing viral infections and things like that. So, our cells will also have carbohydrates, long chains of sugars attached to some of the proteins on the outsides of the cell. Some of them can also be found embedded inside of the phospholipid bilayer. Now, these sugar membranes these sugar molecules, also play a role in cell-to-cell recognition, like I mentioned earlier. The immune system will roll around, it will read proteins, it will also read the sugar molecules that are found on the outsides of the cells to verify that that cell is supposed to be where it's supposed to be, kind of thing. So they're also embedded, throughout the cell membrane are going to be cholesterols. So cholesterols are going to make sure that the cell membrane is not too flexible, it's not too fluidy, it's not too rigid, it's not too soft, not too stiff. So these cholesterols are going to be added or subtracted at a controlled rate to make sure that the cell membrane is not too squishy. You don't want a latex ball surrounding your water balloon that's too thin because it'll pop. You also don't want one that's too thick or too stiff because nothing will be able to come in from the outside of the cell, won't be able to diffuse through. So those cholesterol molecules that are embedded in the cell membrane help regulate that process. So as I mentioned, plants have a cell wall, and that cell wall is going to be found sitting right over the top of their cell membranes. You can see the cell membrane down here, and our cell wall that sits right over the top of it. Now, in the case of cell walls for plants, they're going to be made out of cellulose, a very, very, very long version of sugar, very polysaccharides, a very strong, very water-resistant form of sugar. Now, what this cell wall is going to do is it's going to allow this plant to be very strong. It's going to allow them to stand upright. If you essentially just had a water balloon and you tried to make a tower of water balloons, it would probably just fall over. But if you took the rubber balloons and you wrapped them in paper mache, which is what we're doing with the cell membrane here, it would allow you to stack them much taller. And this is what allows plants to grow upright and stand up, even though they essentially are just little balls of water. And one of the other things that this does is it helps the cells from exploding. You have a nice rigid wall of paper mache on the outside of your water balloon, so you have a much stronger water balloon surface, so you can put much more water inside that water balloon without it popping. You have a bigger, stronger wall. Whereas if you had a little weak rubber wall, just a cell membrane, you're more likely to pop with taking in too much water. And plants need a lot of water. They need to have too much water inside of their cells. And that's what allows them to stand up, allows them to stand upright and not wilt. If you've ever underwatered your plants, you know what I'm talking about. You give them some water and they perk back up again. So that cell membrane and that cell wall work together. That cell wall is what's going to prevent the cells from bursting. So let's talk about the organelles that are involved with the eukaryotic cells. Now, first things first, we've already talked about the nucleus, how important a nucleus is for cells and what that does to break apart our prokaryotes from our eukaryotes. Now, inside of the nucleus, you're going to find DNA. And now, the nucleus is a little protective layer of phospholipids, another bilayer that's going to surround the DNA of an organism, all of the collective DNA inside of an organism. Now, all that DNA is going to essentially function as the recipes to make proteins. So, you want to make sure that the cookbook that's used to make the dishes, aka you, the cell, stay very safe. You want to make sure that they don't get broken apart. You want to make sure that they're protected, and that's what the nucleus does. It protects the recipes. It's essentially like putting your cookbook inside of a safe. You want to make sure that it doesn't get damaged, it doesn't get destroyed, it doesn't get broken down. So that's what a nucleus does. It just protects the DNA of a cell and keeps it safe. Also inside of the nucleus, you're going to find a little ball of RNA and proteins and things called the nucleolus. And the nucleolus is going to produce ribosomes, and that's where ribosomes are going to be made. Ribosomes, you can see them on the outside of the cell, on the outside of the nucleus. They're going to be producing proteins. So what happens is the ribosomes are going to be produced inside of the nucleolus. They will be produced inside of the nucleolus. They will migrate out of the nucleolus through the nuclear pores here, the little holes inside of the nuclear envelope, out inside of the cell. Some of them will bond to the endoplasmic reticulum, which we'll talk about in a second, and some of them just float around in the cytoplasm. So let's go ahead and move forward. So once we have our DNA here, we're going to take our DNA inside of the nucleus, and that DNA doesn't make a protein. We're going to have to take that DNA, and we're going to have to transcribe it into a language that can be read by ribosomes. Ribosomes can't read DNA. Ribosomes are going to make our proteins, but they can't read DNA's recipe. So we're going to have to rewrite that DNA message into RNA. So this one's called mRNA. There are multiple types of RNA. We'll talk about those later in this lecture series. But this one is called mRNA, or messenger RNA. Now, this messenger RNA is essentially just DNA in a language, the DNA recipe in a language that the ribosomes can read. So what's going to happen is this is all going to occur inside of the nucleus. This DNA is going to be transcribed into mRNA. That mRNA is going to also be protected by the nuclear envelope. And that mRNA is going to leave the nuclear envelope through the nuclear pore here, you can see that there, and venture onto a ribosome. Now, once that mRNA reaches the ribosome, or before it reaches the ribosome, it has to leave the nucleus. And I talked about that just in a second. So here's our nucleus here, the nucleolus in the middle. It's going to leave the nucleus through one of these little nuclear pores. And you can see these little holes inside of the nuclear membrane. So anyway, once the RNA is going to leave those nuclear pores, it's going to bond to a ribosome. Those ribosomes, as I mentioned earlier, are going to read the mRNA message, and they're going to prepare the dish, the protein, from that recipe. And they're going to be the chefs that make the recipe, which is pretty cool. So that's very useful for cells. You need to be able to make proteins. Well, most ribosomes, not all of them, are going to be floating around, going to be attached to the endoplasmic reticulum. The endoplasmic reticulum is essentially just an extension of the nuclear envelope. You can see that here. It's just a bunch of folds of the nuclear envelope with the ribosomes stuck to it. It makes sense that your factories that make proteins are going to be stuck to the place that makes the right recipes for them. It just comes right out of the nuclear pore and right on into the rough endoplasmic reticulum, and you make a protein right then and there. It makes a lot of sense. Whereas some of the ribosomes are going to be floating around in the cytoplasm of a cell. So if they're floating around on the inside of a cell, they're going to just have an mRNA or something bond to them there, and that protein will be transcribed in the cytoplasm. So once this happens, once the mRNA is going to bond to a ribosome, essentially what's going to happen is you see our ribosome here. This is the bottom part of the ribosome, top part, and the mRNA is in the middle. And what's going to happen is the mRNA is going to be pulled through the ribosome, and the growing protein chain is going to be added on the other side of the protein. This is the one that's floating around inside of the cell's cytoplasm. Now the other side over here is if it's attached to the rough endoplasmic reticulum, the protein, instead of being made and floating around in the cell's body, the cytoplasm is just going to be made and put inside of the endoplasmic reticulum. And when it's made inside of the endoplasmic reticulum, it can then be altered and moved around, once again, significantly easier than having to go round this guy up and go alter him. And they're going to be made in different places due to their functionality and where they're needed to be used inside of the cell. So let's go ahead and talk about what's going to happen next in this process. So once our mRNA has been made, we're going to have our protein, and we're going to enter into a different process that deals with proteins, that moves the things that the ribosomes have made around. This is going to be our endomembrane system. It's going to contain the nuclear envelope, our endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and our cell membranes. So this process is talking about making milk. So let's talk about what happens here. So what's going to happen is all the different membrane parts are going to work together to make proteins, which will eventually move stuff outside of the cell. So we talked about how our mRNA is going to be used to get to our ribosomes, and those ribosomes are then going to be used to make proteins. The proteins, you can see here, are produced and are going to secrete those proteins in order to make milk for the organism that's eventually going to drink the milk. So all of these proteins are going to be produced inside of the cell, and they're eventually going to have to leave the cell to enter the milk ducts where they can used to make milk. So what's going to happen is all of those proteins are going to move from all the ribosomes into the endoplasmic reticulum, where they're modified and changed around and gotten into the correct shape that they need to be, added and moved and altered and changed to where they're supposed to be. They're then going to move into the Golgi apparatus, and you can see the Golgi apparatus here. The Golgi apparatus is then going to package up all of the altered proteins and move them out of the cell, move them where they're supposed to go. It's going to send them out of the Golgi apparatus to the cell membrane, if that's where they're supposed to go, to the milk ducts in this case, if that's where they're supposed to go. It's going to send them where they're supposed to go. And how it does that is it packages up the little proteins in these little tiny membrane-bound balls called vesicles. And these vesicles are essentially just a little ball of phospholipids with a bunch of proteins and stuff inside of them. And those vesicles will carry the proteins from the Golgi apparatus to the cell wall, our cell membrane in this case, and then out of the cell membrane. That vesicle will bond to the cell membrane. You can see that here. And eventually it will leave the cell. Okay, so let's talk about what happens with the organelles that are involved with breaking down energy inside of the cell and breaking down waste products and breaking down food. So what happens when the cell is going to be damaged or the cell eats something that needs to be broken down? So something useful, something for nutrients. So the cell, what's going to happen is the cell is going to find some nutrients. It's going to bump up against something. You can see that here is on the surface, on the outside in the environment. So it bumps up against the cell's membrane. Now what's going to happen is that cell is going to suck in its cell membrane and form a lysosome on the inside. It's going to, you can see here that lysosome is going to contain, or lysosome, sorry, is going to contain all of the debris, the nutrients and junk and things that were sucked up from the outside, as well as some digestive enzymes, some things that break down organic molecules and things on the inside. So what's going to happen is all of that food is going to be broken down by those enzymes inside of that lysosome. And then once it's all broken down, the lysosome is going to be broken apart, and all of the nutrients can then be absorbed by the cell, which is pretty cool. All the waste products that are left over can then be repackaged up in another lysosome and then sent out of the cell. Lysosomes can also be used to break down damaged cellular parts like mitochondria, things like that. So if something inside of the cell gets broken, you can send a lysosome to that broken organelle. The lysosome, as you can see here, has broken down that damaged mitochondria. It's eaten the mitochondria on the inside, it brought it inside the cell, and then it's going to break it down. No point in having broken cellular machinery floating around inside of your cell when you could break it down and recycle the nutrients inside of it. So most plants, as I mentioned earlier, are going to have that big vacuole in the middle of them. And this is one of those key differences between plants and animal cells, other than the cell membrane, or cell wall, and those chloroplasts. So in plants, cellular digestion is going to occur inside of that big vacuole. This is where they're going to store water. You can see that here. This is where they're going to regulate enzymes and store salts and things like that. So this vacuole does lots and lots and lots of different things for a plant cell. Very, very, very important. So this is where its cells are going to break down. Plant cells are going to break down all of the good things that they eat and bring inside of their cells. So lots of cells, animal cells in common, for the most part, have things called peroxisomes. Now, peroxisomes, as their name implies, contain hydrogen peroxides or some sort of peroxide enzymes that are used to break down organic materials, fats and things like that, sugars and stuff, breaks them down, proteins. So these peroxisomes are essentially just little balls of vacuoles, or if you like, lysosomes, of little balls of peroxides. So what's going to happen is the immune system uses these a lot, and they contain toxic chemicals. Peroxides are mostly toxic. So if the immune system finds a cell it doesn't like, or something bad inside of your cell happens, these peroxisomes can be sent. It's essentially like throwing a poison water balloon at a bad guy. So the cell will send out these poison water balloons to attack invaders and things that are trying to take over a cell. Or trying to kill you, things like bacteria that are trying to harm your cell, or something that's gotten inside of the cell that's dangerous. It will try to break it down with those peroxisomes to try to prevent the cell from getting any further damage or dying. So that's pretty cool. So now let's talk about how cells get energy. You're probably familiar with the mitochondria. Mitochondria are the powerhouse of the cell. Mitochondria are very interesting. They have their own DNA inside of them, which is very strange, a very unique DNA. It's called mitochondrial DNA. It's inherited from your mother. So you can trace mitochondrial DNA back for generations, which is really, really interesting. It does not change very much. These guys also have their own ribosomes. It's a different form of ribosome than a prokaryotic cell, which is also very strange. Something if you guys get if you want to look up if you want to go down this rabbit hole, look up the endosymbiotic theory. Very strange. We may speak about that in a little while. Anyway, the mitochondria is the powerhouse of the cell. And what's going to happen is it's going to be where cellular respiration is going to occur inside of the cell. And this is where cells are going to take the energy from food and break it down and change it into ATP that then can be used to power the cell. So cells will only run on cellular gasoline, ATP, and the food that we eat is going to be broken down by those cells and turned into ATP. And this is where that's going to occur inside of the mitochondria. So you can see here that the mitochondria, the space inside of it, is called the matrix. And you can see that here inside the mitochondria is called the matrix. And inside the matrix is a really, really, really folded up membrane called cristae. And this is where those cellular respiration reactions are going to occur. They're going to occur inside of the matrix and inside the surfaces of the cristae. And this is where cellular respiration is going to occur. Now, chloroplasts are essentially plant versions of, now some plant cells have mitochondria, but these are essentially the plant unique version of a mitochondria that uses light to make energy instead of food energy, which is really cool. So chloroplasts are very specialized structures that are found only in plant cells that allow them to take sunlight and convert that along with water and carbon dioxide into sugars that plants can then use to convert into ATP that they can eat, which is very, very, very cool. So this is how this one works. Very strange. So inside of a cell, chloroplast is called the stroma, and the stroma is going to contain large stacks of membranes called thylakoids. Now these thylakoids contain photosynthetic pigments. That's where photosynthesis is going to occur. So the light's going to come inside from the outside, penetrate through the cell, hit this chloroplast. It's going to hit the thylakoids. The thylakoids are going to undergo photosynthesis, and all the energy inside of this chloroplast is then going to be converted into ATP and then used to power the cell. So very, very, very cool process that only plants and a couple of protists can do. The sugar that's made in here is then going to be transported out of the chloroplast to the mitochondria, where the mitochondria inside of those plant cells will then change that glucose into ATP for the cells to use. So they have mitochondria as well. They can just make their own sugar in these chloroplasts, which most eukaryotes, which us, unfortunately, we cannot do. So the last part of this is the cytoskeleton for the eukaryotic classifications. So the cytoskeleton, as the name implies, is essentially going to support the inside of the cell. It's going to allow the cells to move around. It's going to allow organelles to transport. It works kind of like the road, and it's going to aid in cellular division. So you can see the cytoskeleton here, all these long purple strands that sit right along the cell membrane or underneath it. So the cytoskeleton supports and keeps the cell membrane rigid and keeps it open. So the cytoskeleton in a eukaryotic cell is made up of 3 different types of filaments: microfilaments, intermediate filaments, and microtubules. And you can see them down here. A microtubule is a tube, microfilaments are intermediate filaments, and microfilaments, 2 tiny little actin molecules versus larger complex proteins. So you can see the difference here. So microfilaments are composed of actin filaments. Actins are proteins that also make up our muscle filaments. Intermediate filaments are going to be made up of proteins. They are not all the same. It just depends on the type of intermediate filament. Different types of proteins are going to make up different types of intermediate filaments. And our microfilaments, on the other hand, are going to be made out of tubulin, which is a very interesting type of protein. It's put together in a unique way to make that circular structure. Now, all 3 of these different types of filaments are going to be interwound together, interconnected. They all work together to form the cytoskeleton of a eukaryotic cell. So, microtubules make up things like the cilia and the flagella that are used to move cells around. So microtubules, you can see here, this is a sperm cell, and the flagella here, the long whip-like structure that allows this cell to swim around through the environment. It's going to be made up of long chains, long strands of microtubules. Those long microtubules are all going to twist together, flip together, flop together to allow, in coordination, to allow this sperm cell to swim. Over here, our cilia are going to be made up of the same things, long strands of microtubules that are going to allow this cell to swim along through the environment, or in the case of our respiratory tract, it works kind of like a dust. It sweeps things up through our esophagus, through our bronchial tubes, and through our respiratory tract. It's called the ciliary escalator. And this is how you, when you swallow dust or you inhale dust particles and things like that, these little guys sweep it up through your respiratory tract and try to prevent it from going into your lungs. So really cool functionality on those. And these are made up of microtubules. So that's pretty much all on the inside of the cells and how they divide up labor and what the specialized structures and functions are on the inside of each one of those cells. Cells all have to talk to one another, and that makes sense. If you don't talk to the cells next to you, you won't be able to coordinate the body movement properly or coordinate growing and reproduction properly and things like that. So if you're a multicellular organism, you have to coordinate to make sure that that organism grows as it should at the right time, and that all of the body parts are being supplied with the right nutrients and things like that. So organisms that are multicellular, their cells are always talking to one another. Lots of energy is used to communicate with cells, other cells, the cells surrounding them, to make sure that the body is in constant homeostasis, everything is happy, everything's good, to make sure that nothing needs to be changed so that this organism can survive. Now, unicellular organisms is a little different in these guys. They don't really have to communicate so much. They don't really need organisms around them. They can live by themselves quite fine. But they do have the ability, and they do quite regularly, talk to other organisms that are in their same species. Maybe it's easier to live together in a colony. Maybe this blob over here of bacteria found some food, and let this blob of bacteria over here know. And that's a nice little thing that they can do sometimes. Hey, come over here and there's lots of food. So they do communicate with one another because, once again, they all are the same species. They all do want to survive, kind of thing. And it's better as a collective species to survive than to fight against yourself, kind of thing. So once again, cells can communicate with one another, and plant cells do this a little differently. Plant cells, since they have that rigid cell wall, and they're all connected, you can see up here, they have this very thick cell wall. Sometimes there are little holes inside of their cell membranes, their cell walls, that connect the cells to one another. They're called plasmodesmata, and you can see that here. It's a little channel that passes through the cell membrane and the cytoplasm from one plant cell to the other. And you can see that here. It goes right through, and you can see where the little plasmodesmata are. And this allows plants to transmit chemicals, signals, and things like that, as well as nutrients from one plant cell to the other. And this is very, very, very important for plants, because if the roots aren't functioning well, you also need to let the roots know if the leaves are damaged and things like that. So this is how plants communicate with one another, by sending these little chemical signals and nutrients and things through the holes in their cells called plasmodesmata. Animal cells will stick together. Sometimes this is how we form our multicellular organisms. You have to be able to stick together as a cell to be able to have multicellular organisms that live together to form one large organism, kind of thing. The cells have to be able to stick together. Now, the different types of sticking allow for different scenarios to arise in different types of tissues. So some cells, when they form together, this is going to be the cells of our small intestines here, and you can see down here they represent all three of them. They're going to form something called a tight junction. The two different cells, when they butt up to one another, are going to be very, very, very tightly connected to one another. This forms an impenetrable barrier, impermeable barrier, where nothing can get through these two cells. There's nothing that's going to be able to squeeze between them. It's going to be very strong. There's going to be nothing that's going to break these two cells apart. But the problem with that is they're not very flexible. So this particular type of tissue is not going to flex very much. It's going to be very strong. There is also something called an anchoring junction. And an anchoring junction attaches the cells together on the outside. It's essentially like holding out your hand and your neighbor holding out your hand, and you grab each other's hands and hold on to each other's hands. It's not quite as strong as if you were glued to your neighbor, but still a pretty strong bond. But what it does is it allows some flexibility. You can flex the person that you're holding hands with can flex. You can kind of see that down here. And that allows some movement and some flexing to occur. And that's a trade-off that you get. It's not quite as strong, but it's a little more flexible. So the type of tissue, depending on what you need it to do, might have more anchoring joints than tight joints. And then you can find gap junctions. And this is when cells have small holes that connect them to the cell that's right next to them. And this allows cells that are very closely related, that have other types of joints and things holding them together, to just talk. They can send nutrients, they can send molecules, ions, and things, hormones and stuff through these little gap junctions to talk from cell to cell. So eukaryotes, as we've talked about, they're very specialized. They divide up the labor to get different types of things done. But since the different types, even the cell itself is specialized, but even the organisms that eukaryotic cells make up tend to also have very specialized tissues. We have different types of eukaryotic cells that only do one thing. We have eukaryotic cells that only function as muscle tissue, that allow us to have muscles. You have eukaryotic cells that are hair cells, that are eye cells, that are liver cells, and they only do that one type of thing. Now you've got plant cells, you've got root cells, you've got wood cells, nerve cells, brain cells. All those different types of eukaryotic cells do very, very, very specialized things. And when you take a bunch of specialized cells that all do the same thing, you get something called a tissue when you stick them all together. So if you take a bunch of specialized eukaryotic cells that are all muscle cells that only work to do muscly things, you stick them all together, you get muscle tissue. Same with brain. You take a brain, a bunch of neuron cells, nerve cells, brain cells that are all the same. You stick them together, you get a brain tissue. You get wood, take a bunch of wood cells, you get wood tissue, plant tissue, leaf tissue, things like that. And that's what tissues are. It's a very specialized grouping of very unique specialized eukaryotic cells. So, even though these cells look drastically different, they are all eukaryotic, but they're all specialized to do different things that are required by the organism itself. So our muscle cells, as you know, allow us to move around. They transmit electrical impulses from cell to cell and allow us to be able to respond to the environment and stimuli around us using these nerve cells. Whereas leaf cells, you can see up here, even though they're both plant tissues and eukaryotes, this is a photosynthetic cell. It's green, it has lots of chloroplasts. You can see them all inside. Whereas our root tissue, which is not photosynthetic, does not. It has no green inside of it. They're both eukaryotes, but they're all specialized to do different things, and that's the point here. Evolution leads to some very interesting outcomes, very specialized functions. So once again, guys, this is the end of this lecture. If you want to look over the appendix here, this will tell you about all the photos and things like that, what's going on in the photos. But if you have any questions at all or you'd like me to elaborate on anything extra, please feel free to send me an email, and I will be more than happy to do so. If not, have a great rest of your day.